For decades, clinicians used percussion tests to determine if an implant was ready to bear the load of prosthetic. They would strike the implant and listen. A high pitched crystalline sound suggested a tight bond with the bone, while as a low, dull thud meant it was loose. It was entirely subjective, relying completely on practitioners' ear. Modern implant dentistry replaced that acoustic guesswork with biomechanical data. The global standard for this is resonance frequency analysis or RFA. RFA system outputs a specific metric, the implant stability quotient, a proprietary scale running from 1 to 100. Clinicians rely on strict thresholds. On this scale, a score of 57 ISQ correlates mathematically to 150 micrometers of micromotion. The physical limit where implant can remain stable enough to achieve osseointegration. Surgeons swapped subjective hearing for digital screen. The ISQ score became the assumed universal standard for deciding whether to proceed with immediate loading protocols. Within the industry, the ISQ value is widely treated as an absolute physical constant, but empirical data reveals a strict contradiction. If you measure the exact same bone implant interface using three different RFA hardware devices, you receive three distinct numbers. In clinical practice, this variance dictates whether a patient leaves the chair with a new tooth or is told to wait several months for the bone to heal. The data shows that an implant deemed perfectly stable and ready for a crown by one device might be flagged as a critical failure by another. Treating the ISQ as an infallible metric while ignoring the hardware generating it introduces a systemic hidden risk into the surgical loading decisions. To see why the devices disagree, we have to look at what they are physically measuring. RFA works by emitting magnetic pulses from a handheld probe to excite a transducer peg screwed directly into the implant. The magnetic pulses cause the peg to vibrate, and these induced micro vibrations reveal the resonance frequency of the entire connected implant bone structure. The structural mechanics dictates how this works. This equation mathematically links the interface stiffness directly to measured frequency. As trabecular bone hardens and heals around the threads of implant, the overall structure becomes stiffer, driving the frequency upwards. But the RFA devices do not display the raw physical frequency in kilohertz. They run that frequency through a proprietary translation to generate the 1 is to 100 ISQ scale. Because the ISQ is merely a translation of the physical resonance, the mathematical algorithm inside the specific hardware becomes a critical variable. A 2021 ex vivo study published in Applied Sciences tested how much this hardware variance affects the final score. Researchers established a standardized physical matrix by surgically placing 30 identical implants into fresh pig mandibles. They then measured stability of those implants using three distinct commercial devices: the Osstell beacon, the Penguin device, and the MegaISQ device. To ensure comprehensive data, methodology required testing each implant in two orientations, taking measurements both parallel and perpendicular to their transducer axes. The control assumption is straightforward. Scanning a standardized identical physical interface should yield identical ISQ scores across all testing hardwares. Any statistical deviation in the resulting numbers isolates the hardware architecture itself as the sole cause of variance. The data was evaluated using intra-class correlation coefficients or ICC. This measures reliability, how consistently a device returns the exact same number upon repeated testing. The Penguin device achieved the highest intra and inter-rater reliability scores. It was the most consistent tool in the test, but the statistical consistency does not equal true device accuracy. A device can consistently repeat a systematic bias when analyzing variance against the mean. The Penguin device exhibited a distant systematic bias, overestimating the ISQ by 4.2 points during parallel measurements. The MegaISQ showed the opposite bias, consistently underestimating the ISQ by 2 points. The study also noted that inter-reliability universally plummeted across all three devices when measurements were measured perpendicular to the peg. The hardware testing proves that highly consistent device can still deliver the data that is artificially inflated or deflated. We can apply these hardware biases directly to the rigid biological threshold of 150 micrometers of micromotion. Consider a hypothetical clinical scenario with borderline implant. It is true physical stiffness equates exactly to a 55 ISQ. If a clinician measures this implant with the Penguin device, the 4.2 positive bias yields a passing 59 ISQ. This clears the implant for an immediate loading protocol. If that exact same implant is measured with the MegaISQ, the device underestimates the stiffness, yielding a failing 53 ISQ. This triggers a delayed loading protocol. The biology didn't change. The decision to load the implant or leave it to heal was dictated entirely by an arbitrary hardware selection and its invisible algorithmic bias. The underlying mechanical engineering of these tools forces them to produce divergent data sets from their same physical input. The physical parameters vary. The different devices use different transducer metallurgy, with some systems relying on standard aluminum smart pegs while others use magnetized multi-pegs. The software parameters vary as well. The proprietary closed source mathematical algorithms that translate kilohertz into final ISQ differ widely by manufacturer. The physical and digital individual variables compound upon each other. Differing hardware architectures and proprietary codes alter the interpretation of raw physical resonance before the clinician ever sees the number. The ISQ is not the universal constant, it is highly specific, device dependent quotient. Clinicians cannot mix hardware. Comparing a baseline surgery reading from the Osstell device to follow up reading from Penguin device is statistically invalid. The correct clinical application of RFA relies entirely on the delta, the relative change in the quotient over time. Tracking this delta requires using the exact same hardware and the exact same parallel measurement orientation for every clinical follow-up. By understanding and controlling for hardware bias, the ISQ ceases to be deceptive absolute number and functions as a calibrated relative indicator of osseointegration.